So she hasn't connected to the audio.
Oh, okay. Yeah, I can just see the. Anyways, okay. Let me. Then she also won't be able to hear me, right?
Yeah.
Let me just write to her. Um, I sent a message to everyone, right?
I
Could you,
didn't get the message.
uh, could you see my message in the
No,
chat?
sir. No.
Okay.
It might be personally to her. That's.
Yes, Abhi has done. Hello, Abhi. Am I audible to you?
Yes, sir.
Yes. Good evening. How are you?
Good evening, sir. I'm good.
Okay. So since this syllabus is completed, we are going through some important stuffs that I have planned for revision. And if you have any doubts, we will first take those doubts. So we are starting with Fatima's doubt from biotechnology regarding uh, regarding the cloning site, I think, and selectable markers, right, in the plasmid. Okay, so we by now what we have studied. Let me give you a quick recap to both of you. Okay, and Fatima, let me know if these things are clear to you first.
Yes, sir.
So we studied that the most important two core techniques in biotechnology on which the whole field is based on is one is genetic engineering, where the techniques that are used are directly to alter or modify the chemistry of the DNA, the genetic material. So we are engineering the genetic material itself, right? So that's called genetic engineering. Sometimes also we do not need to change the genetic material. We just need to do bioprocess engineering, where biotechnology still will help us with techniques like IVF. So in IVF, like in vitro fertilization, we do not tweak or change or modify the DNA of the sperms or the ovum of the gametes in general, right? But we create a whole process, which is again a biotechnological process to make these gametes fertilize outside the body. That's why it's called in vitro fertilization. And once fertilization has happened, then we need to put it back into the female's womb for growth, right? The embryo is transplanted back. So here you will see that there was no genetic engineering as such, but that also comes under biotechnology. A very simple thing like growing a bacteria in specific environments which do not allow any other kind of contamination to happen also comes under bioprocess engineering. So bioprocess engineering has much wider applications in every process of biotechnology. Okay. Now we will first focus on genetic engineering. So to genetically engineer a DNA, what do we mean? We just mean that we are making a recombinant DNA enter a host cell and replicate and express itself, because we know that DNA contains genes, and these genes can be read by transcription machineries inside the cell, and then translational machineries that convert genes into proteins. Correct? That is
Yes,
clear?
sir.
Clear? Cool. Now the idea, the concept is how to achieve this feat, how to make a recombinant DNA. So DNA, what we have studied is in our cells or in general, DNA is a very long polymer, right? A long thread spooled together, compacted, fold, coiled, super coiled to make chromosomes, right?
Yes, sir.
So how to use that molecule to to carry a gene of our interest to any host cell that we want. Now, the problem with linear DNA is that linear DNA is more susceptible to degradation by enzymes called DNases, right?
So what is linear DNA?
Linear DNA is which has a beginning and an end. So a linear chain. So just one more. So this, our DNA, like a human DNA, is just like this, right?
Yeah.
It's a long thread. It's a linear DNA, right? So linear DNA has an end and a beginning, where sometimes I've told you that human chromosomes towards the end and the beginning, of the thread have a very long repeat of, most of the time it's a similar kind of repeats, AAT, GGC, this kind of repeats multiple times, thousands of times. So these are called telomere repeats. Telomere repeats protect the actual information of the DNA. So I mean to say that, let's say from this point to this point is your genes. Okay, there are multiple genes on the DNA which are helpful to you, but DNA ends here. So you will continue to make some more repeats. So these do not code for any genes. These,
So
yes,
this is a linear DNA. So is there a possibility of an infinite DNA? No, right? All DNA ends at some
right,
point, and
right. And
begins
then,
at some point. So why do we call it linear DNA and not just normal?
yeah, linear means normal. Linear simply means a straight chain, straight. Circular means like this: linear, circular, and then coiled,
Okay.
right?
Yeah,
So it's
yeah.
a linear, straight DNA. It can open up to have two ends. Anything that has two ends is a linear thing. So we want to protect the ends. So at the ends, we continue to make some DNA repeats. Okay. Now these DNA repeats are just for protection. They are called telomere repeats. Now with age, let me tell you this interesting thing. So with age, it has been shown that telomeres keeps getting short and short. And if they become shorter than a specific length, they are not able to protect the DNA properly. So that's why age-related disorders, diseases, loss of DNA happens. And that is one of the things associated with aging. So people think that if we can protect our DNA from our telomere from shortening, we can maybe reverse aging. But then they did that and they realized, oh, that's not the only factor that causes aging. That is one of the factors. So also in cancerous cells, it has been shown that um, telomeres stay short only, but still cancer cell keep dividing. So if telomeres are short, then your cells should not survive, but cancer cells survive with a short DNA, short telomere. So many things. So it's all multifactorial, but one
How
of the,
does that happen?
sorry.
How does that happen? Because cancer cells divide, and then there are so many.
Yeah, because cancer cells are just what do you say? They are rogue cells. They do not follow the normal cell biology.
Oh yeah.
So you know that normally when a cell divides, it has to go into a resting phase where it grows, right? You cannot
Yes.
immediately after one cell division start another cell division, right? But cancer cell can do that. And also normally when cells come into contact with each other, there is a property of contact inhibition. That's how our body structure is maintained. Whenever there is a wound in our body, and wound means, injury means the cells from that region are lost, cut or scratched away or lost. So you have a wound. Now that wound will heal. But every time with with a very great precision, our skin heals and does not overgrow, right? It
Yeah.
just heals the amount of loss it is bearing. And whenever all these cells are regained back, divided back from the stem cell niche, it stops dividing further. Body is healed. So something is telling the mechanism that okay, mm, the hole in the membrane is filled, the gap is filled, so stop dividing. That property of contact inhibition is not present in cancerous cells. So they actually just continuously keep dividing to an extent where they just invade the nearby tissues also. Okay, physically they can just make a tumor and invade and go inside, you know, nearby tissues and organs and cause damage to those as well. Anyways, so we were talking about linear DNA. So using a linear DNA is not a good idea. We cannot use that, and it has other problems that I will tell you about in a minute. So we decided, let's find what we can do. So we already know about something called plasmid DNA in a bacterial cell. In prokaryotic cells, there is a concept of plasmid DNA, right? And you know what is plasmid DNA? Fatima, would you like to?
So it is a DNA in the bacteria which does not replicate.
No, it does replicate. Otherwise, what is the point of DNA if it does not replicate?
But
In
it
fact,
doesn't form proteins, right?
it does form proteins. It has genes, it replicates, form proteins, and that is what give bacterial cells a lot of advantage.
So what is
yeah,
the difference between plasmid DNA and the genomic DNA? Because then
the genomic DNA is the bacteria's own DNA. It's the genome. Genome means what constitutes all the gene that bacteria has right from the beginning.
but the
Plasmid DNA,
plasmid is
plasmid
also
DNA.
there. Sorry.
Yeah, sorry, no, you're right. But the plasmid DNA, bacteria takes from the environment. It is capable of exchanging plasmids, taking up any foreign gene and incorporating it. So plasmid is a circular DNA. It is autonomous replicating DNA. That is what is fascinating in a bacterial system. That if a bacteria, so there are many bacteria in nature that have evolved antibiotic resistance on their own, right? We didn't do we didn't teach them how to be antibiotic resistant. They just became antibiotic resistant, all of us, all by themselves. So that happened because through mutation, and we also know that bacteria cannot sexually reproduce, right?
Yes.
But bacteria have some mechanisms which are equivalent to sexual reproduction in terms of variation. Bacteria can generate variations. So if one bacteria, in order to survive, underwent any mutation, and that mutation helped the bacteria to somehow avoid antibiotics or degrade antibiotics, or one common thing is, so what happened is there is one bacteria. So the antibiotics that we give to bacteria are of two kinds, first of all. So I'm digressing a little, but to understand you this concept of foreign DNA, which is different from the genomic DNA of bacteria. So every bacteria, when we give antibiotics, there are two kinds of antibiotics. One is called bacteriostatic, one is called bacteriolytic. Bacteriostatic antibiotics do not allow bacteria to grow further, but it doesn't kill the existing bacteria. Do you understand? Sometimes all we need is to not allow them to grow. If we cannot kill the bacteria, don't allow them to divide. You understand?
Yeah.
So if they don't divide, and if you have a mild bacterial infection, it will stay mild for many days, and our immune system, macrophages, will finally find all of them and eat. Done. Job is done, right?
Yeah.
The other way is, this bacteria is obnoxious, we have to kill it. So we just kill it. That's called bacteriolytic antibiotic that kills the bacteria. So most of the time, the bacteriolytic antibiotics have to enter the bacteria inside. Okay, and they enter it through bacterial pores. So bacteria have different channels and pores that it uses for exchanging nutrients and substances in and out of itself, because bacteria have to also cooperate and interact with its environment. Every organism has to, correct?
Yes, sir.
So we take advantage of those channels, and we know that this channel can put my drug inside the bacteria. So I put my antibiotic that goes inside the bacteria in flux and kills the bacteria. So due to this antibiotic, so now this antibiotic is like a stress to bacteria. Yes or no?
Yeah.
So what is bacteria? An organism. So when we give stress to bacteria, that stress acts as a natural selection stress for that species. So there will be mutations happening. And by chance, one mutation helped the bacteria to reverse, the ability to reverse this channel. So the same channel, bacteria started making copies in a reverse order. So we are putting antibiotic inside them. They're using the same channel in reverse order, just adjacent to it, to put the same antibiotic out. That's called antibiotic efflux. And this is one of the mechanisms that bacteria acquired, and our antibiotics, they hence don't work now.
So how can you kill a bacteria?
Then we find other ways to attack the cell wall, to attack some other pathways. So that, there are many pathways, glycolytic pathways. If I stop glycolytic pathway somehow of just specifically the bacteria, which is very, very difficult, because our cells also have the same pathway. But bacteria, the one thing that our cells don't have is cell wall. So most of our antibiotics attack the cell wall, because if I have a drug in my body that is attacking the cell wall and breaking down the peptidoglycan layer, it will never harm my own cells, right? Because my cells, human cells, have no cell wall and no peptidoglycan. Make sense to you?
Yeah.
But then the problem is, if we can't make a specific antibiotic, after antibiotic doses, our own good gut bacteria also die, a lot of them, because those will have a cell wall, right? So there is a
Yes.
cost that we have to pay, but we get rid of infections, of course. So this inversion mechanism,
So if the other bacteria die, then how do you compensate for them?
we replenish them. So we allow, again, you eat, because those are good bacteria. When you eat food, a lot of them come from the food, like curd, milk, probiotics. So sometimes doctor also advise someone who has underwent a bacterial infection plus a diarrhea kind of a symptom, then they say that, okay, you should take some probiotics now. So those probiotics have just good bacteria, gut bacteria, which we call. So these good bacteria are important to help us with synthesizing some very crucial micronutrient elements like vitamin B12, etc., etc. Right? So we replenish them. We give time to again come back, these bacteria to grow. So if you look, it's very interesting that now research is coming out from mice models where they have figured out that the gut bacteria, we used to think that gut bacteria, we are the host, gut bacteria is the parasite, so it lives, and over time it made a mutualistic relationship with us, that you benefit me, I benefit you. Okay, anything that came first might have just invaded, right? Our bodies were not understanding, or our cells were not knowing the bacterial cells to begin with. But now we cannot imagine ourselves without bacteria in our body, to such an extent that it's not just crucial, it actually, our moods, our hunger cycle, our cravings to eat something different, all of it depends on our gut bacteria to some extent. Even our sleep and wake patterns. Now that was very, very interesting, a study that came out in mouse models, that if you perturb these bacteria somehow in the mouse model, then their behavior also changes. So we all humans have different concentrations of different kind of gut bacteria. Okay, mostly we all might be having a similar pool of gut bacteria, but maybe in my gut there is something more than which is not in your gut. And it also keeps changing with day and night. At the daytime, our gut bacteria concentration is different. At the night, it is different. And that is governed by food we eat. So it's amazing. So if I look, sometimes I think that we say that we are the most evolved organisms, and we are the dominating ones. But what if we are the inferior organisms serving these bacteria? What if they are just using our bodies to just survive and live? And if you go back to the theory of endosymbiosis, these are just like across the lines thoughts. If you go to the theory of endosymbiosis, you know that one bigger bacteria swallowed a smaller bacteria but did not kill it. And that prokaryotic bacteria could actually tolerate oxygen, stress, and convert and use oxygen to convert to make energy. right? Which earlier, oxygen is not a good thing for a living system. That's why we have to use a organelle called mitochondria in our body. If you remove mitochondria from all human cells, we will die because of oxygen. Do you know that, people?
No.
Yeah. So oxygen came out as a stressor, as a toxic stress in the environment to living systems. There was an event in evolution called Great Oxidation Event, GOE. If anyone is interested, can just read about it. It happened when photosynthesizing bacteria and algae started evolving, and there was lots and lots of sunlight available, lots of oxygen could be produced because lots of CO2 was there in the environment. It was reducing environment at that time. So you just take CO2 and you do photosynthesis. And you take water, lots of water was available. You split the water, use the hydrogen to reduce the CO2 and make starch, and then oxygen is a byproduct. So oxygen is actually a waste of photosynthesis process. And they started doing that, and lots of oxygen was being pumped in the atmosphere. And atmosphere all of a sudden started becoming oxidative. And all the life by then was not very happy with oxygen, because oxygen is a very, very reactive molecule. It does combustion, oxidative phosphorylation, oxidative breakdown of substances. Even in our body, we use oxygen for the very same purpose. We use oxygen to destroy things. We don't use oxygen to make things in our body. We use oxygen to do oxidative breakdown of glucose and produce energy from it. So oxygen is like a sword. Now imagine if we do not know how to control it. At that point, what happened was, archaebacteria. There are three kinds of. So the problem is that NCERT does not teaches you about domains of life. Now we are a little digressing, but is it okay, Fatima, if I take some time to
Sure.
tell you these things? Abia and Matthew, because these are doubt and concept classes, so it will help you for your entrances. How are you, Matthew?
I'm fine, so I just came back from my face-to-face classes, which I had.
Yeah, okay. So we're discussing about, basically we had to discuss about recombinant DNA, but we went back a little in time, in our time machine, and we're discussing oxygen and glycolysis. Is it okay?
Yeah, sure.
Yeah. So what was I saying? I was saying something. Sorry, I'm very bad at this.
Something about oxygen.
Yes, but what about
Domains of life.
Yes, domains of life. Perfect. Thank you so much, Matthew. So we study kingdoms, right? Plantae, Animalia, Monera, Fungi, Protista. Before kingdoms, there is a concept of called domains of life. All these 5 kingdoms come from 3 domains. Domains are archaea, bacteria, and eukarya. So there are some kind of bacteria which does not fit well with bacteria. They are called archaebacteria. Your NCERT just talks a little bit about it in one small paragraph, I think in 11th class, if I'm not wrong. So archaebacteria, and they call the normal bacteria as eubacteria, which is true bacteria. And then eukaryotes, of course. So archaebacteria are very, very interesting because we thought they were bacteria, but they are not bacteria. In fact, if you look at their structure of the cell wall, their cell wall is different from bacteria. Of course, eukarya, only fungi and plants have cell wall. Animals don't have cell wall. But the theory of endosymbiosis makes more sense after discovery of archaebacteria, and they are called extremophiles. So archaebacteria can live in extreme of environments, hot geysers, the most saline water body, and very, very high pH, high temperature, I already said. Even colder temperature, archaebacteria have even been found in ice, right? Now that may, all of a sudden, the theory of evolution, early evolution of life started making more sense that, oh, so these are the ones which are the ancestors of all life. And these are the ones which used to live before the great oxidation event in very, very extreme environments, because Earth was not very pleasant at that time. It was not a pleasant heaven to live in. So these were the bacteria. Now what happened? Suddenly oxygen came, and all the oxygen producing cyanobacteria are the true bacteria. They are not archaebacteria. So these true bacteria evolved, and they started utilizing oxygen and also producing oxygen. Now it became a problem for archaebacteria. But what they did, one archaebacteria must have engulfed a bacteria, but instead of killing it, they just somehow evolved into a mutualistic relationship, because true bacteria, it was difficult for them to find a safe shelter, because archaeas were always hunting for them. And for archaebacteria, it was very difficult to sustain in an oxygen rich environment. So what happened, this true bacteria, when entered inside the archaebacteria, it got shelter, and this started helping the bigger cell, the archaea, with oxygen stress. And now we know that smaller bacteria that got inside in eukaryotes as mitochondria. Now again, sounds like a very fancy story, but where is the proof, right? It could all be just coming from a madman's mind. Someone just sat one day and made all this story that there was one big bacteria, one small bacteria, it swallowed the small bacteria and things like that. But the point is, now we have because now we have technology. So when people sequenced mitochondrial DNA, a human mitochondrial DNA, they thought that the mitochondria of human, which is a eukaryotic organism, should also be eukaryotic in lineage, right? You know that prokaryotic DNA is different from eukaryotic DNA. You can just know it by looking at the sequence, because there is something called a concept of GC content, guanine cytosine richness. This is studied in, this is one marker. Another marker is, in a eukaryotic genome, you will find like spaces which does not code for non-coding regions. I told you, right? 98% of our DNA does not code for proteins. And when we got to know this, we were like mind boggled. What? Only 2% of our DNA codes for proteins, which is like around 28,000 genes. That's it. Humans just have 28,000 genes. Butterflies have more genes than humans. So people started discovering things that did not fit. Then they realized, okay, on the basis of GC richness and other regions, looking at this coding and non-coding region, we can actually figure out what is a prokaryotic DNA, what is a eukaryotic DNA. Then we sequenced mitochondrial DNA, and we realized, wait a minute, mitochondria does not have any eukaryotic lineage. Mitochondrial DNA is very similar to a bacterial DNA. Was mitochondria a bacteria at one point of time? Yes, we already had a theory proposed by someone who said that, archaea cell once engulfed a eubacteria, which went into a symbiotic relationship and started helping the cell with oxygen stress. Yes, it makes sense. Mitochondria helps us with all the oxygen stress in our body. That electron transport chain where oxygen is reduced to water takes place in the mitochondrial inner membrane. Yes or no?
Yeah.
Can you join dots now? Right? So this is how we started using oxygen for our benefit. Okay. And still there are many archaea that cannot do that. So they live in anaerobic conditions. So that's what about how a new, and also mitochondria has its own DNA. So that is not the only extra chromosomal DNA in eukaryotes. There is also something called plasmids, which are there in, plastids. I'm so sorry, not plasmid. Plastids, which are there in plant cells, right? Chloroplasts, leucoplasts, amyloplasts. They also have their own DNA. And they might also have been coming from a similar lineage. So a lot of evidences in plants is also being gathered that plants have mitochondria, plus they have plastids as well. So how it happened there, what happened there, people are still researching. A lot of answers we have, a lot of proofs we have, but that is not the point we want to discuss here. Similarly, a bacterial cell, when shows small mutations, okay, in its own genome, which allows the bacterial cell to mitigate or to counter any problem, like this problem of antibiotic resistance. So what happens when a bacteria dies? It has been already shown by Griffith. So Griffith's transforming experiment, if you remember. Do you remember that, Matthew?
Yeah.
What was that? Would you like to? I'm connecting dots from very different parts.
So like there were So he did on two mice where there was the R strain and the S strain
Of what?
of DNA or like the
Yeah.
genetic material used.
No, no, no.
Pneumonia.
Yes, it
Oh
used
yeah. Sorry,
it.
yeah.
He used Streptococcus pneumoniae bacteria. Mice was just a model on which study was done. People always remember mice, but they don't remember Streptococcus pneumonia. That was the most fascinating thing. So what happened?
Yeah, like a weakened R strain, um, when it was like introduced into the S strain, like form essentially into the more
Mm, okay.
cautious.
So Streptococcus pneumonia is known to cause pneumonia. It's a bacteria that causes a disease. It's a pathogenic bacteria, right? Okay.
Yeah.
Now, Streptococcus pneumonia had two kind of strains which were discovered. One is called the S strain and one is called the R strain. And very interestingly, only the S strain is virulent. Only the S strain causes pneumonia, the disease, and kill you. R strain does not cause the disease. Now, why is that first? Because it was seen that why is S strain called S strain? Because the S word stands for smooth. So these bacterial colonies these bacteria make very shiny and smooth colonies. And that shininess or smoothness comes from the fact that these bacteria have a gene that can produce a mucus, a mucosaccharide, okay, on the surface. And you know that mucus is slippery. If your cell wall is coated by a mucosaccharide, you will be slippery and shiny, right?
Yeah.
So these bacteria are slippery and shiny. R strain is rough. It looks rough on the plate. If you culture these bacteria, they will look very rough colonies. They will not shine in light or something like that. They are not slippery because they cannot produce that uh, that molecule on the cell wall. So somehow this gene is associated with causing virulence in the cell. Okay, so this gene, this mucosaccharide, is actually important for interacting the bacteria cell with our lung cells and causing this disease, getting into the cell and multiplying there and infecting the lung. So if you inject S strain into the mice dies, of course, within a week. Makes sense? Because S is virulent. But if you inject R strain, the mice lives. Nothing happens to the mice. Now, what he did, he heat killed the S strain. So if you boil the S strain bacteria, the bacteria is dead, right? Upon boiling. And then you inject the heat killed or heat weakened, what Matthew was saying. When you inject heat killed S strain, what do you expect? Mice should live or die? Abia?
They should live.
Mice should live, right? Because it is a heat killed bacteria. So mice lived. Hooray, you all are genius. Then he thought, okay, let me do one thing. What will happen if I inject a heat killed S strain bacteria and a R strain bacteria together? Remember, when you do that individually, when you inject heat killed S strain separate, mice lives. When you inject R strain, mice lives. So none of these are capable of causing any disease in the mice, and the mice lives. But to the amazement, what the interesting part was, when he injected a cocktail, a mixture of heat killed S strain, which is the lethal strain, and live R strain, which is again not lethal anyways, right? The mice died of pneumonia. What happened there? No one. So there was a puzzle. This was when they were trying to figure out proofs for what is the genetic material. So they were puzzled. Okay, why is that happening? So this is what Griffith's transforming principle is. He proposed that there is something which got transferred from one strain to the another strain, the dead strain to the live strain, that made that live strain transform into a lethal strain. So R strain by itself cannot harm, but when it was injected with a dead S strain bacteria in its vicinity, something from this passed on to that. Now, what was that something? That was a puzzle at that time. Okay, but you realize that what is happening here? The mice died. Interesting, right?
Yes, sir.
So he realized what is happening is bacteria are capable of taking foreign DNA. So when you heat killed S strain, the bacteria died, the cell wall bursted, the DNA got broken down into multiple pieces. All are lying here and there. But this specific gene, which causes this S strain to be S strain and make this mucopolysaccharide, this was taken up by the R strain randomly. It must be floating around, this gene, and this gene was taken up by the R strain, incorporated in its own DNA, and then the R strain transformed into S strain. Now, this is all a story. You have to prove it. So what will be, how will you be convinced? What should I do to prove this to you? If I inject dead S strain plus live R strain in the mice, then the mice dies. Then I cut open the mice. And after cutting open, I take the blood or the lung sample. And from that lung sample, if I can isolate live S strain bacteria, which I can culture on the plate and show to you that, see, this is a S strain live bacteria, will you be convinced that the R got transformed into
Yeah.
S? Great. So this is exactly what Griffith did. And he found that in an organism where we have not injected any live S strain bacteria, only live R strain was there. From that, after its death, you can recover live S strain bacteria. So which means there could be transformation by exchange of genetic material. Now, what was the genetic material was later found by Hershey and Chase in bacteriophage experiment very beautifully. But this bacteria can do, this is what is important here. So you know that bacteria can take up extra chromosomal DNA also, extra genomic DNA also in their cells. That is what plasmid DNA is. And if it is extra genomic DNA, how will it be stable and safe inside the cell? So for that, it has to be circular, because circular DNA cannot be attacked by exonucleases, okay, from the outside, or the enzymes that damage DNA or start chewing the DNA off. So because it does not have loose ends, a circular DNA is more stable and protected than a linear DNA. So that's why this plasmid DNA is circular. Coming back, does it make sense now, Fatima, Abia, and
Yes,
Matthew?
sir.
Perfect. Now, this plasmid DNA can contain gene, of course, and it does contain gene. That's why bacteria has taken it up. And it is autonomous. It can replicate independent of the genomic DNA. Okay. And it can be replicated, transcribed, and translated independent. So whatever gene is there in the plasmid DNA, most of the times in naturally occurring bacteria which have plasmids, the genes which are there on the plasmid provides the bacteria with additional benefits in its environment. The ability to tolerate temperature, the ability to fight off antibiotics, the ability to find food better, the ability to metabolize some special kind of a food, etc., etc. So we realized, okay, now coming back to making recombinant DNA, this all the knowledge helped scientists, and they thought, okay, so it means you can pack something in a plasmid DNA and send it inside the bacteria, and bacteria will replicate, transcribe, and translate it in its cell. And and interestingly, it can even transcribe and translate a gene that is not a bacterial gene, even if it is a eukaryotic gene that has no advantage in bacteria, because plasmid is autonomously replicating. You understand? It does not have to be in the control of genome. So it will happen. So we just started. So humans are always opportunistics, right? We are opportunistic. We said, okay, let's do it. So let's isolate plasmid from bacteria, naturally occurring bacteria. We isolated a lot of plasmid, and we realized there are a lot of genes in the plasmid. We're like, okay, chuck all the genes. Yes, Fatima.
Why did we not do this for the genomic DNA?
Because genomic is very, very big. It is genomic DNA. It's like if you take out genomic DNA from a bacteria and you want to push that back into a bacteria, it's not possible. Bacteria will not take the genomic DNA. There can't be two genomic DNA in the same bacteria, right? It can only take some small fragments. from
Yeah,
outside.
if you take its own genomic DNA
Mhm.
and then you modify it and then put it back inside,
But how will you put it back inside? That's the problem, right?
how you put the plasmid?
We just allow the plasmid to be present in the environment. Then we heat shock. A small pore opens. Plasmid goes inside. Plasmid is small. Plasmid is sneaky. That is the whole point of doing biotechnology, no? You culture bacterial cells. We make bacterial cells competent by treating them with divalent cations, calcium chloride in this case. Remember, I taught
Yes.
you all of that.
Yeah.
And then we just give them a heat shock. We don't do anything fancy. You can send your plasmid inside the bacteria just by putting them on ice, chilling them, and after chilling, just put them on 42 degrees Celsius. And when you are doing that, in that tube, in their environment, there should be the plasmid, your plasmid present. So what will happen? These pores in the walls will open and plasmid will just enter inside. It is a small thing. And your gene, because all you want is just your gene to enter, right? It could be one single gene, any gene that you want to make the protein of. So that's why there's no point taking out genome and then we cannot put back the genome inside the bacteria unless and until we do it for one single bacteria where we inject it through a micro injection. But that's also not possible for bacteria. Not possible. I should not say not possible. I should say not feasible technically. Bacteria is very small. It is possible for a sperm or a ovum cell, but not for a bacteria.
Yeah, even for putting the plasmid, we use gene gun, right? That's why
Yeah,
I was thinking we could do that.
again, but what you're saying is plasmid is like a bullet, right? And we use gene gun on those cells which have a, like, mostly for plant cells. Okay, because plant cells can suffer that injury and heal themselves, even for bacteria sometimes, but no, for bacteria there is a very easy way. Put them on ice, put them on 42 degrees heat, they will take it on their own. So why shoot them with guns and all, right? Gene guns and all. And the concept of gene gun is also that it is a very small nanoparticle of either gold or tungsten on which your extra chromosomal gene, the gene of your interest, will be coated. And when that small nanoparticle like a bullet will enter inside the cell, your gene will also enter with it. And then the cell will heal itself and the DNA is inside, it will get translated and your product is ready. So we use gene gun or biolistics for plant cells mostly because they have a very, very strong cell wall, which is different from bacterial cell wall and which does not respond to heat shock. So we have to resort to violence a little bit in scientific terms. Okay, I hope you understand that, right?
Yeah.
Yeah. For animal cells, we use microinjections, not gene gun, because animal cells will die. They don't have a cell wall, or if their plasma membrane is damaged beyond a point, they will die. Bacteria have a cell wall, and which can open and close in heat shock because they have a heat shock proteins for that. So we just are using knowledge from different domains, putting it to make something that we want to make. Is it clear till here, everyone, the process?
Yes, sir.
Yeah. Yes. So that's where we take plasmid. We remove all the unnecessary genes from that plasmid. And how do we do it? By DNA cutters called endonucleases. And then we put three things in the plasmid, which are very important. One is ORI. Either the plasmid should have it naturally or you put it if you are designing a plasmid. ORI is called origin of replication. This should be there because that decides how much the plasmid will be replicated inside the bacterial cell because it is autonomous. If you do not have ORI in a plasmid, that plasmid will not replicate. Okay, Fatima? So the purpose is
Yes, sir.
failed. So we put ORI, then we put the gene of our interest, let's say insulin, because I'm interested in making insulin. Okay, I want to be rich, let's say. So this blue will be the gene A or gene of interest, which is insulin next to ORI. And rest is the plasmid that does not, we don't care what, it should not be just any gene that hampers insulin. It could be anything else. But third important thing is the selectable marker. Now, selectable marker, you talked about selectable marker, right?
Yes, sir.
Selectable marker is a gene that we are putting inside the plasmid, which is not useful. So we don't want that gene product to be isolated. Okay, we just are putting that gene so that we can know which bacterial cells are transformed, which means they have our plasmid, and which do not. Because there is a possibility when you give this heat shock treatment, chemical heat shock treatment, to make the cells competent, not all the cells will become competent, right? It is a natural phenomena. So not all bacteria can take up plasmid very well. And in fact, in nature, only 1% of naturally occurring bacteria that we have found till now can naturally take up plasmid from outside. The rest 98 to 99% bacteria do not take up extra DNA from outside very easily. Might be they have not evolved yet. So we have to push them to do this. So when we push them, chances are maybe they will die in the process and not take the DNA. Maybe they will live through the process and not take the DNA. And third chance is they will take the DNA. So the ones that are died. That's not the problem. But the problem is to identify between those which have taken our DNA and are alive and which have not taken our DNA and are alive. Because in that tube, you have all kind of bacteria. When you are growing them on the plate and when you are growing them in big vessels, if you are growing the bacteria which does not have your plasmid, it's a waste of a process. At the end, you will not get anything from them, right?
Yeah.
So we don't want that to happen. We only want to select the bacteria that has our plasmid. But how will I get to know which bacteria has my plasmid? I cannot see the plasmid. Insulin cannot be seen when it is produced inside the cell. So there has to be something which can be seen, through which I can detect. So we use a gene which acts as a selectable marker. Now there are two ways to use selectable marker. One is mostly, which is the most common way, is to use a antibiotic resistant gene here. So this selectable marker is an antibiotic resistant gene. Let's say this is a gene against ampicillin. Okay, ampicillin is an antibiotic, right? So if my bacteria, E. coli, let's say we are starting with the E. coli. Now we know that E. coli cannot survive in antibiotic. It does not have its own antibiotic resistance. So if it has taken my plasmid, my designed plasmid, my recombinant DNA, then it has now a gene in itself, which is a ampicillin resistant gene. So now after doing this competency thing, what I will do, instead of plating them on a normal plate, I will plate them on a nutrient plate containing ampicillin. So only those bacteria will survive which have my plasmid. You understand? Because I have given the gene, ampicillin resistant gene.
Yeah.
So in that way, this gene that I introduced in my plasmid became a selectable marker for me. On the basis of this, I can select which bacteria have my plasmid and which bacteria does not have. So those who will not have my plasmid will die. So let them die. I will only culture those which have my plasmid, and the best way to culture is culture in a medium where ampicillin is present. At the end, all the bacteria that I will be sure that they have my insulin gene because they are showing survivability in ampicillin. So ampicillin resistant gene is a selectable marker here. Is it clear, everyone?
Sir,
Fatima?
if ampicillin resistant gene is put in inside,
Mhm.
then how will you differentiate insulin?
Because it's in the same plasmid I have designed. My insulin, ampicillin resistance is like a proxy to insulin. So you have a best friend and you both always stay together. And I am using your friend as a proxy. So if I can see your friend in school, I will say that, okay, you are also in the school because you both are connected together. So you can see that the insulin gene is next to the ampicillin gene.
Yeah.
So if bacteria is producing this and getting resistance, it also must be producing this also because it's in the same plasmid under the same origin of replication. It's not possible that it will jump gene A and only produce this. That's not possible, right?
Yes, yes.
So you understand? So that's where I'm using ampicillin resistance gene as a selectable marker. So that's one way.
One more thing. See, this is a plasmid and it will produce so many proteins. How do you divide the proteins? Like,
So many means you mean to say there are also so many genes here.
Yeah.
Yeah, I can design that. I can remove anything that is a gene that produce proteins, and I can put gibberish sequence. So most of this plasmid has gibberish sequence, no gene, which are designed by humans.
So
In
this
nature,
plasmid is completely made by the humans, or it is taken from the bacteria?
you can take it from bacteria and let few genes, which are bacterial genes, or genes which are helpful for bacteria, be there. For that matter, the genome also produces a lot of proteins. Your question is how at the end, after culturing, I will isolate insulin.
Yes.
Yeah, for that there are protein isolation techniques. Every protein has some different fold or domains, and it interacts with something. So for that matter, insulin interacts with insulin receptor, right? So I can just, after at the end, take all that millions and trillions of bacteria, crush all of them so that all the cytoplasm comes out. Now I have a soup of bacterial cytoplasm, and I know that there must be lots and lots of insulin protein in that soup. All I have to do is to purify, to just tag and remove insulin, but no other protein. That's called a protein purification process. For every protein, you can use a bait which will pull off, like protein will bind to that and other proteins will just wash away. That's how you purify any protein. So protein purification in itself is a very big field of science that we have developed. We can purify all proteins. So when your blood goes for blood test and they look for some protein levels, how do they do that? Same thing. In your blood, they will use the concept of antibodies, right? Anything which is a protein, you can make an antibody against it. So we can make an antibody kind of a molecule against insulin that only binds to insulin and nothing else. So all the insulin in the solution will be bound to it, and you can purify it. And then after purification, you make a resuspension in some chemical that keep the insulin stable. And that comes as an insulin shots, and you take it in the injection and put it inside the diabetic patient. So all the insulin that comes for diabetic patients are formed inside bacteria these days. Okay, make sense?
Yeah.
So I was telling that this is one way to use selectable marker. One way is to use antibiotic resistant genes as selectable marker. The other way is, which is called insertional inactivation. I think I gave this, yes, as a homework. Did you read insertional inactivation? So there we don't use antibiotic resistance gene. Instead, we use this one gene which codes for an enzyme called beta galactosidase. Now it is a ACE, which means it's an enzyme. Now what does this enzyme do? If this enzyme is present and active, it will react with some molecules present in its vicinity. So if instead of antibiotic resistance, if we have used beta galactosidase as a selectable marker, so now we have to check on the basis of color on that plate. So if beta galactosidase is there, which is in our plasmid, then there will be blue colonies formed. But if I inactivate beta galactosidase, now what happens here is we have to design the plasmid a little different. So this is a plasmid. Look carefully. And this is your ORI, okay? And then this is your insulin gene, okay? Sorry, no insulin gene till now. Sorry, forgot. Yes, ORI, and this is your beta galactosidase gene, okay? Let's say it's a big gene. Now, in this kind of plasmid preparation, we first cut the beta galactosidase and put our gene of interest in between beta galactosidase. So this is our gene of interest. This is our gene of interest X, and green is the beta galactosidase. So if my gene is present, beta galactosidase will be inactive. Make sense?
Yes, sir.
Yes.
And if my gene is not present, beta galactosidase will be active. Make sense?
Yes.
So If my gene is not present and beta galactosidase is active, then it will form blue colonies because beta galactosidase will be produced. It will react with something and form blue color in the solution in the bacterial cells. If my gene is present, it will inactivate beta galactosidase and the colonies which will be formed will be colorless. So if I get colorless colonies and colored colonies on the same plate, I will know that which colonies to pick and culture further, the ones which are colorless. This kind of, and here the good thing is we are not using any antibiotic, because antibiotic resistance also comes from misusing or overexposure. So people thought that, okay, is there any other way we can do this? So yes, you take a gene and you insert your gene of interest in between that gene to inactivate that gene, and then see somehow if you can identify these two different kind of colonies. So this kind of mechanism is called insertional inactivation, because we are inserting our gene of interest in between the beta galactosidase gene and inactivating this beta galactosidase gene, on the basis of which we are getting two kind of colonies, and then we can select our colonies. So that also works as a selectable marker. Make sense? Fatima, Matthew, and Abhi?
Yes, sir.
Perfect. And then comes to cloning site. Cloning site is nothing but in the DNA, foreign DNA, which is the plasmid, which we have created, there has to be recognition sequences which restriction enzymes can recognize and cut at. Those are called cloning sites, because wherever in plasmid we are making a cut, there is only one reason to make that cut. And what is the reason? Why are we cutting plasmid?
To insert gene of interest.
Perfect. And why are we inserting DNA of interest? To clone it.
To make a protein
Yeah.
of the
Recombinant.
desired protein.
Basically, to clone it, right? To make the gene clone.
Yeah.
Make multiple copies. So the site at which these restriction enzymes cut the plasmid are called cloning sites. Now, for one restriction enzyme, would you or would you not like to have more than one cloning sites in your plasmid? Do you think that more the number of cloning sites for one restriction enzyme, the better it is?
No.
No. You know why it is not better?
If too many cuts will be there, then it will be very difficult to manage it.
No, it's not
No,
about
it will be
managing.
very hard for the restriction enzymes to cut it.
No, restriction enzyme can cut at 1 lakh positions if you have 1 lakh sites.
Yes, sir, but then you might not get it at like the right area, right?
No, you will get it at the right area because they are very, very specific. That's the power of restriction enzymes. The problem is not that restriction enzymes will be confused because of too many places, or it will be too difficult to manage. The problem is exactly the opposite. If there is more than one cut, a circular thing when cut more than once becomes linear. That is the main problem.
Yeah.
We always want the plasmid to be cut at one side, insert our gene. So this is the plasmid. You cut it here, it became linear. Then you inserted your gene.
Structural integrity.
Yeah, you can again make it circular easily, because whatever you will insert in between will be ligated at both the ends. But if you cut here and you cut here, so you will have one fragment this and one fragment that. Now it will be difficult to make it linear again, right? Unless and until you are inserting two genes, which is again more complicated process, and we lose the plasmid the moment we cut it more than once. From a circular DNA, it becomes linear, and linear DNA is not stable. It can degrade faster inside the cell. So that is the whole point. We only want one cloning site for one restriction enzyme. We can have multiple cloning sites, each cloning site for a specific restriction enzyme. That is there. That is exactly what your NCRT shows you. If you go to your NCRT, there will be a picture of PBR 322 plasmid, which is one of the most commonly used plasmids in the field. Can you open your NCRT and find this picture, PBR 322 plasmid? Everyone?
Yes.
Let me know when you find that. And please share the page number for others, whoever finds first.
So page 199.
page
199.
199. Come to page 199, people,
I
in your
got
textbooks. You got the PBR 322 plasmid
yes.
diagram. Can you see there are many endonuclease sites shown here? BAM, H1, SAL, HIN3, etc., etc.,
So
PSL, etc. So these are all restriction enzymes. So all these multiple restriction enzymes have have exactly one site where it can cut in the plasmid. Now this is called a preformed plasmid. You can shop for plasmid these days. So labs would work on. So in my lab, we just ordered for one plasmid because we are trying to clone a gene in Drosophila in fruit flies. So we can just order plasmids. We just tell them, see, this is what I need in my plasmid. They will prepare it for you and give it to you. Now you have a variety of options to choose that which restriction endonuclease I want to choose to cut my plasmid, because all the sites are already available there. So it acts like a very convenient tool these days to tweak genetics and all. Okay. Make sense, people? Any other doubt with cloning sites or plasmids, vectors, anything?
so why can't we? Can we use radioactivity for as a selectable marker?
We can, but radioactivity causes cancer. So too much of exposure to radioactivity can also affect the experimenter. So radioactivity is not a safe option. When there was no chemical tags or the chemistry was not evolved, biochemistry was not that advanced, people actually did use radioactivity. Marie Curie worked her whole life on radioactivity. Marie Curie is the only female scientist who has been awarded Nobel Prize twice, right? Even Hershey and Chase, they found that DNA is the genetic material by using radioactivity. You can use that, but difficult thing is to handle it, to keep it stored, and radioactivity causes DNA damage. So if the experimenter is, so if I give you an option to go for radioactivity or to go for chemical plasmids, what will you choose?
Chemical plastic.
Exactly. Okay, so earlier, if you see all these kind of experiments which were done 50, 60 years, 70 years ago, they were all radioactivity based. Scientists were just, what do you say, enthusiastic just to figure out the truth, did not care about what's, of course, they took a lot of precaution. It's not like they just handled radioactivity with their bare hands. There is a way to work with radioactivity. Even it's not like it has gone out of science forever. In chemistry research, radioactivity is used because chemists work with radioactive elements. People are trying to make new radioactive elements for new kind of researches. And all of that is done by wearing protective gloves, gears, and all and all. For plasmids, you need just gloves, sanitized, and a clean, aseptic environment. That's all. So works very handy. But yes, theoretically, we can use that, Fatima. Good question. Okay, others? Doubts? We still have time. So more doubts from any chapter or from this chapter? Anything else you want to discuss? So the better you will understand science, the better you will perform in both entrances and boards. So don't just remember these question and answers like literature and go and write it. And I hope all of you will end up pursuing a career in some domain of science, maybe as a doctor. Physicians also need to know a lot of science. That's the problem. When you go to grad school, you will realize that the medical schools do a very good job, and that is the point of becoming a doctor, and teaching you pathology, physiology, how disease works, everything. But they don't do that much greater job to teach you how are you treating it actually. So when you become doctors or when you are becoming doctors, always keep in mind that don't just prescribe drugs to patients because prescribing that drug will give you more commission from a pharmaceutical company. So even to prescribe paracetamol, there are many ways: Crocin, Dolo, XYZ brands. Always look for the composition of the drug. always know or try to learn how that drug is functioning. I have seen people who are physicians, great meritorious physicians, doctors, done MBBS properly. If you ask them, how does this drug going to work in my body, they don't know it. They just know that it's going to cure your vomiting, nausea symptom. Now I understand that it's not possible for one person to know everything, right? But at least some basics. So doctors are specialized to treat and cure. So if you pursue that career, the most noblest of the careers on the planet, it's good to be doing that with some backing of fundamental science. And similarly, many scientists don't know like whether to go to doctor or not during what kind of a disease. So the problem is equal on both sides. I think knowledge gap can fill that. And a lot of fundamental science is now being done with doctors. So there are an increasing number of people now who have done their MBBS, and now they are doing PhDs because they are clinicians who want to become clinical scientists. And who can understand disease better than a doctor? No one, right? They have a first-hand experience of seeing a cancer tumor, treating a cancer tumor, surging out a cancer tumor, or let's say goiter or any kind of a disease, leprosy, skin rashes, skin disorders. Now, when they come to the lab, sit on the bench, try to find a cure, they will have a notch edge advantage over those who have just studied it in their BSc, MScs and have come for PhDs. So at many places in the world, there's a program called MD PhD program, where you do your MBBS, and after your MBBS, you go to do a combined MD and a PhD, for which hospital will be funding you, because the hospital knows you are an asset. You are MBBS. If they invest something in you and you you find something, a cure, or during your PhD, MD, PhD, you will come back to the hospital, solve the hospital or the field better. So in India, first of its kind program is started by, I forgot the place, ISC Bangalore or ISER Pune. There's something from these two. It has started a MD PhD program. It is first of its kind in India. It's available in other countries. So it's a good thing. So the future ahead of you, my friends, is very bright, because we are going to be attacked by lots of pathogens, viruses, bacteria. They are not defeated. We might be looking like victorious in this pandemic war because we survived and COVID, but COVID still is surviving and boiling, planning its next move. And not just COVID, many others are. So we need people like you. Make sense? I hope
Yes, sir.
I motivated you a bit to see your entrances. Yeah.
Very like not
Sorry?
bit.
Yeah. So to see your entrances and your career choices and your studies, future studies, don't just see it as a mundane thing you have to do in this society. People just study because studying is a norm in society. If you are illiterate, society will like, oh, he's illiterate. But when you're studying, study with that zeal. When you're giving the entrance, you should know, okay, why I'm giving the entrance? Because one day I'll do this. I'll contribute, right? So take it as a positive, enthusiastic pressure, handle it well, emerge victorious. And let me tell you, there is not only one way to be victorious, to go to a medical school or to go to a PhD. There are many ways to be victorious. Now you know about biotechnology. You can be a biotechnologist. People do biotechnology after they do engineering. And there is an engineering program in biotechnology. It's called BTech Biotech. So those are engineers who are engineering biology. They are not engineering bridges and buildings. They are engineering molecules and cells. So those are also called engineers. The degree is a BTech degree. There's a MTech in biotech as well. Yes, so
That's
many things. Sorry, yes, Fatima?
That's very interesting.
Yes, that's very interesting. Great. So anything else you people want to discuss? So since the syllabus is over, I wanted to discuss few terms with you. So we are trying to, because now many students who were just who are not continuing because the syllabus is over, so they wanted what they got, and it's completely understandable. So I've delivered what was promised, but I also promised that if you want to stick with me, I'll stick with you. So this continuation of these classes will depend on till the last student wants to continue before the boards. Okay, and I already told you, after your boards are finished, during the boards there will be no classes because it makes no sense, because next day you are going to give a physics board. I should not teach you biology a day before. So after your boards are over, the one and a half month of gap, we will again organize a crash course for just entrance oriented mindset. Okay, but before that, till your boards come, I have planned to go through certain chapters, topics, and take some previous year's questions from NEET exams and solve it. So these will be the major outlook of the classes. So the idea is from next week, we are joining weekend batch to make one batch. Okay, which will be, so currently you join at what, 5 KSA, 5 p.m. KSA,
Yes,
right?
sir.
You join. So that batch, which is the on weekend, from next weekend onwards, will be a combined batch of yours batch and the batch after you. 2 batches which I take on weekend will be combined into one, and it will be called a concept builder batch. Till board it will continue. Till the last student is willing to come, we will continue that. Okay. So this last stretch is not at all profit oriented. It's actually our motto to find students who can, because if you will do good, and yes, there is a selfish motive, and that is, if you will do good and someone will ask you who taught you biology, you'll say my name, right? I hope you do that. I want to be remembered. Anyways, but since the last one, till the last one of you wants to continue, I'll be continuing these classes. But from next weekend, it will happen at 6:30 KSA, which is 9 p.m. Indian time. Is it okay? Will you join at that time? Are you willing to join at that time?
Same time, right?
6:30.
6:30.
6:30 KSA.
6:30 KSA. Because we are merging it just for those students who want to discuss doubts, concepts, etc. Now, what is your opinion?
Okay, sir.
Yes,
I
Matthew.
don't mind. I don't mind.
Are you up? Abia
Yeah.
and Fatima, what about you?
Yes, sir, no problem.
No problem. Abia, okay, cool. So we have 3 S's already from here. So you 3 will be, so we'll continue it next weekend as well. Okay, cool. So let's decide a theme. So next weekend, let's start with some, because reproduction is a very heavy unit, right? Both for boards and for entrances. So how about we, yes.
Genetics is more important, right?
Genetics is also very important, but reproduction is the base of genetics. So next Saturday, let's take reproduction first. Okay, major important topics and questions, and then next Sunday we'll take genetics. How about that?
Yes, sir, no problem.
Yeah, so there is a theme, so come prepared with your doubts from reproduction unit at 6:30 p.m. KSA. Okay, and bring all your doubts, and if you have no doubts, then the way I have planned it is I will first go through my notes, all the important things. I will ask someone, because along with you there'll be a couple of or some more students from other batches, so it will be a good inter-batch interaction to see. And one thing which is very honest is, like, I find this batch, which is your batch, which is MED, it's a very, very vocal batch and very discussive batch. So that's a advantage that you people have. So a lot of things that is discussed with you, which is actually initiated by you. So if Fatima and Matthew and Abia do not bring the kind of questions they bring to the class will not go in that direction, right? Because I cannot decide what extra to tell you, because I know so much extra. If I keep telling extras, we will not be finishing syllabus. But you have to initiate something